Well, thank you very much, Lars. It was a terrific invitation and introduction, that's for sure. And I'm delighted to be here. And I'm delighted to talk about my favorite thing. I wish I could lecture about something other than schizophrenia, but it's really my only topic. And did I understand that I'm going to be on YouTube? So finally, my children will be proud of me. I mean, I don't know if you all have trouble with your children looking up to you or not looking up to you, or say, well, Mom, what do you do anyway? If I'm on YouTube, I'm really going to have made it. So I'm delighted. Thank you very much. So what I'd like to talk about today is schizophrenia, or what I will talk about today is schizophrenia. And I'm going to start out just by talking a little bit about clinical aspects of the illness, and then I'm going to talk a little bit about my focus in the last number of years in schizophrenia has been the medial temporal lobe and declarative memory function. So I'm going to talk a little bit about declarative memory function. Then I'm going to talk about a new model that I've put together in order to test the molecular basis of abnormalities in the medial temporal lobe that we've discovered using imaging and some human postmortem studies. So I'm going to, this is a very speculative model, albeit testable, so I'm going to be very interested in what you guys think about it when I'm at the end of it. So I'm a new learning and memory model of schizophrenia. So in the United States, we always have to put these disclosures about where we do advising. And unfortunately, I don't own anything. I only advise people. And I think it would be better if I owned and didn't advise. So I'd like to talk about schizophrenia for a little bit. These are two of my favorite pictures about the illness. The picture on the left, of course, you might realize as being an engraving from 1750. This is from a hospital in England called Bethlehem Hospital. But when a Brit says Bethlehem, it sounds a little bit like Bedlam, and the word Bedlam came into the English language as a description of the kind of behaviors you see on the left. And that was more or less how I think schizophrenia or psychosis might have looked like in 1750. Then we fast forward to 1996, and although there's really less movement and there's less posturing and perhaps less catatonia, other than that, schizophrenia looks fairly similar as it did in 1996. Is there any chance we could turn the lights off a little bit? I think you could see, if we could turn, if anybody knows how to turn the lights down, the pictures would look a little bit better. The picture from 1996 came from the National Geographic magazine, and it was the first article in a major news magazine in the United States that presented schizophrenia as a disease of the brain. So this was really, it's been a while since, it's been not very long since people have recognized that schizophrenia is a disease of the brain. So one of the things that when I talk to audiences who don't know about schizophrenia, and I think most of you do know about schizophrenia, it's always a little difficult to present some experience about the illness so that people in the audience, at least this is before A Beautiful Mind became a movie and before lots of books were written on it, to give people an idea of what the experience of schizophrenia is like. So you all know John Nash. He won the Nobel Prize in 1994 for in economics. He was a mathematician. John Nash is a brilliant guy. I mean, he supposedly has an IQ of 160 or some phenomenal thing. And he got ill when he was 31. He did his Nobel Prize winning work before he was 30 and had his first onset at 31. And to see that somebody who's this brilliant has normal everyday delusions and schizophrenia. His buddies, his mathematical buddies from Princeton, came to see him in the hospital one day. And after he said, I secretly feel that I'm the left foot of God and God is walking on the earth, his friends said, John, you're so smart. John, how can you really think that? And he said, these ideas come into my head just like all of my other ideas. I have no choice but to believe them. And I'm sure that once we get to understand what a delusion is, that this will be a clue that delusions and hallucinations seem like normal thought processes for the people. Ellen Sachs is another one of our brilliant schizophrenics in the United States. She's a person who was on had done phenomenally in school, was in England on a Fulbright scholarship when she had her first psychotic episode. She had the idea already then that if she told people she was schizophrenic or she was having hallucinations and delusions, that would affect her future life and maybe people wouldn't hire her. So she kept it a secret, more or less. She had a close circle of friends. And she kept it, she didn't really tell anybody that she had a diagnosis of schizophrenia. She stayed in England until her symptoms were under control. She came back to the United States, became a lawyer, got a job at Southern California University, and rose up through the ranks, did a phenomenal job in her academic position. And finally, when she got tenure, probably 2 months after she got tenure, she wrote a book. And the book is titled The Center Cannot Hold. And she writes some experiences of her psychosis. Sights and sounds, thoughts and feelings don't go together. No organizing principle takes successive moments in time and puts them together in a coherent way. So it's always, I think, every book I get to read, a first-person account of people with schizophrenia, I read in order to really understand what their experiences are like. So what is this illness? What is the illness? What are the clinical characteristics of the illness? I don't have to tell a group like you what are the clinical characteristics, but I've put them down here in a list just for a background. So the prevalence is about 1% worldwide. The prevalence is the same in almost every country in the world. There are a few pockets of high prevalence worldwide. And probably due to genetic inheritance in specialized areas. But in general, the prevalence is about 1%. It has a very common onset, beginning at somewhere between 18 and 26 years. Now, what this means is really the psychosis onset is that the psychosis starts. Oftentimes, some of the cognitive and perhaps some of the negative symptoms of schizophrenia are in place before psychosis begins. Symptoms are worse before 50. So in some ways, schizophrenia is like a disease of childbearing years. And oftentimes, but not inevitably, oftentimes symptoms wane, positive symptoms wane in later life. There's a profound psychosocial dysfunction. And I think that when we talk about schizophrenia and talk about the hallucinations and delusions and realize that we have antipsychotic drugs that will treat hallucinations and delusions, we forget that even with optimal treatment, still only less than 15%, in the United States anyway, less than 15% are employed, less than 20% ever marry, and less than 5% really recover. And by recover, I mean lose all of their symptoms. The pathophysiology is not known. Dopamine has always been important. D2 dopamine receptor antagonists are our only known treatments. And there's an anatomy to schizophrenia that I'll show you in just a minute. Both genetic and environmental influences are important. The strongest influences are genetic. We've looked for years for a gene for schizophrenia. So when this first started out, we were talking about a gene for schizophrenia. Then we talked about 5 genes for schizophrenia. Then people talked about 30 genes for schizophrenia. So the last paper really talks about 1,000 genes or maybe 10,000 genes for schizophrenia. So clearly, schizophrenia is a complex genetic illness with spontaneous mutations thought to account for a lot of the vulnerability. Prenatal, perinatal events, winter birth, early environment, and now cannabis use, especially early cannabis use before the age of 15, are being identified as risk factors. So one of the new ways to look at schizophrenia that's gradually emerged over the last decade or so is that schizophrenia probably is not, almost inevitably is not a single illness. It's a syndromal application. And what is schizophrenia anyway? It could be composed of families of symptoms that associate with each other. Large populations of people with schizophrenia have been analyzed, and it looks like the area of cognitive dysfunction runs together, psychosis runs together, negative symptoms run together, and the idea is emerging that schizophrenia as a diagnosis may not be identified by any one of these domains, but may be identified by dysfunction in all of these domains. It may have a pathophysiology that's specific to each of these dimensions, and it may have treatments that are specific to all of the dimensions, not treatments that are specific to schizophrenia. As you know, we don't have anti-schizophrenic drugs, we have antipsychotic drugs. And for that matter, we don't have anti-bipolar drugs. We have drugs that treat depression. We have drugs that treat psychosis and things like that. There's a certain anatomy to schizophrenia. And I've listed out a few of the, I wonder if there's a, I'll just use this as a pointer. I just wanted to emphasize, from now on, I'm really going to talk about the hippocampus and the medial temporal lobe, but I don't mean to say that I think that the medial temporal lobe is the only important area in schizophrenia. It's just the area that my studies have focused on, and I would really like to talk about the contribution of the medial temporal lobe and the hippocampus to psychosis in schizophrenia, understanding that there are many other brain areas, particularly the prefrontal cortex and the anterior cingulate cortex, that are undoubtedly important to symptom manifestations. So what I'd like to first talk a little bit about is taking a look at declarative memory function in schizophrenia. So this is a hippocampus here. This is the anterior part of the hippocampus, and this is the posterior part, and this is the way a hippocampus presents itself when we in a postmortem brain. Probably the hippocampus is a little bigger than your smallest finger, and there's two of them, one on the right side and one on the left. Now the hippocampus came most poignantly to medical attention through the case of HM. So and we now know that HM has a name now because he passed away about 2 or 3 years ago. So then his name became public, Henry Meliasen. And so it was about 55 years ago, that HM was a 27-year-old man who suffered from intractable seizures. And neurosurgeons had just learned that if they could identify where the seizure focus was and cut it out, the seizures would stop. The misfortune of HM was that he had a seizure focus bilaterally on the right and on the left. So the neurosurgeons went in, cut out his seizure focus, and he lost all ability to make new memories. So I think that people never understood that brain regions as small as the hippocampus in a whole huge brain would have a function that was that important as memory, declarative memory, making new memories. So it really made scientists very curious, and it really jump-started the whole field of declarative memory function. As time went on, people understood that there were other cases, perhaps not as traumatic as HM, but other cases of hippocampal sclerosis that occurred because of epilepsy or stroke or high fever in children that would demonstrate the same outcome and the same course. So this is really a schematic, if you will, of the hippocampus and the whole medial temporal lobe. The whole medial temporal lobe takes input from the whole brain, from the whole neocortex. So every part of the neocortex, the sensory part of the neocortex and the associational neocortex, sends projections in to the medial temporal lobe, and the entorhinal cortex really funnels all those projections down into a very characteristic one-way projection through the hippocampus and really out the other side. So one of the things that's unusual about the anatomy of the hippocampus and underlies characteristics of its function, and I will propose characteristics of its dysfunction, is that it's a one-way pathway. So all the pathways in and out of the entorhinal cortex are two-way. The pathways between the entorhinal cortex and the subiculum are two-way pathways, so that the target tissue reflects back on the projecting tissue and regulates it to some degree. But the main part of the hippocampus, from the entorhinal cortex to the dentate gyrus, dentate to CA3, CA3 to CA1, is a one-way pathway, where the subfield that accepts the projection does not feed back in order to regulate the part that the region that it receives projections from. Now, the whole function of this hippocampus is to make complex memories, to make declarative memories. So it has to take input from very many sensory modalities and put them together into a complex image. So I walked in this room today for the first time and looked around, and my hippocampus was working very hard in order to take all of the information, the color of the chairs, the organization of the room, the curtains in the room, all of which I noticed when I walked in, puts it all together in a single memory. And the function of the hippocampus is to more or less make a barcode of that memory. The hippocampus does not store all memories, but it stores the barcode or the code that then gets pulled back to resurrect the memory. So there are several things that are very important to making a memory. One, that's probably one of the first steps in making a memory, is called pattern separation. So I had to walk into this room and I had to try to remember, since I've been in Sweden so many times, have I ever been in this room before, or is this a new memory? And it clearly was a new memory. I didn't have to do very sophisticated pattern separation, since this was a very different room than I'd been in before. If this was a room that was very familiar to me, then I would have had to do some pattern completion to decide whether or not I could resurrect a memory from partial input. And I wouldn't have to make a new memory. Your hippocampus has to automatically decide whether you make a new memory or whether you pull something from the past, and whether you pull up an old memory and add to it. And that work is really done. The work in the hippocampus is done between the dentate gyrus and between CA3. And one of the characteristics of CA3 that you'll hear me talk about are these recurrent collaterals. CA3 has to talk among the cells in CA3, the neurons have to talk to each other a lot, so they have a very active recurrent collateral system in order to judge, in order to make memories and to lay them down and store them. Once the signal gets worked on by the dentate gyrus and CA3, it runs rather directly through CA1 and then out and back to the subiculum and the entorhinal cortex. Here is just another kind of rendition of what I just told you, with the entorhinal cortex projecting directly to the dentate gyrus, projecting directly to CA3, which are with a rich collateral pattern, and then projecting out and producing declarative memory performance. So this is a circuit that I'll show you again, hypothesizing what might be altered in psychosis. Now, there are very characteristic abnormalities of the hippocampus that have been previously reported. First of all, the hippocampus is reliably slightly smaller in people with schizophrenia than in normal people. Not very much, less than 5%, but it's a very consistent finding over almost every study. And it's the most consistent reduction that's been really reported in schizophrenia. There's an increase in hippocampal basal perfusion. If you look at regional cerebral blood flow or regional cerebral blood volume, there's an increase in basal perfusion, which is a little unusual for schizophrenia because most functional changes in brain regions are decreases. But then there's an activation defect during declarative memory. So if you give a person with schizophrenia a declarative memory task to do, they activate activity. They stimulate neuronal activity in their hippocampus less than normal people do. And I'll show you examples of that right now. This is a simple novelty detection task where we put people with schizophrenia into a scanner and showed them a series of pictures, some pictures that were familiar to them, complex outdoor scenes, some that were familiar to them and some that were not familiar to them, and we compared them to normals. And this was an event-related design, so that what you see here is what's called a hemodynamic response function. And what you notice is that the normal activation, the normal HRF, is rather robust in the perirhinal cortex and in the perihippocampal cortex, less so in the hippocampus proper. And the blue line is the line for is the response of normal controls, of normal people, of our control subjects. The red line is the response of our schizophrenic volunteers who were on medicine, and the yellow line is the response of the schizophrenic volunteers who were off medicine. And you see that there's a progressive decrease in activation. This is a decrease in activation within the various regions of the medial temporal lobe. And the group that has the lowest activation are the schizophrenics off medicine. Now this is another more complicated task where where the task measures retrieval and generalization. And I won't tell you specifically what the task is, except that in the first part of the task, we teach people with schizophrenia we teach them associations, and they learn them. They're a little slow in learning them, but once they have learned associations between 2 or 3 different things, they'll remember those associations, and when we ask them to retrieve the associations, both the schizophrenics on medicine and the schizophrenics off medicine retrieve just as their performance, their cognitive performance is just as good as the normals. When we ask them to make generalizations with that information, if we ask them to do a logical problem where they make a generalization, the normals are quite good at making generalizations from previously learned material. The schizophrenics on medicine are slightly worse, and the schizophrenics off medicine make generalizations almost like at a chance level. And this is a little bit reminiscent of the clinical, what we do clinically where we ask schizophrenics to interpret proverbs. This would be a mental task that's very similar to proverb interpretation. And if you look at the imaging that accompanies it, during this part of the generalization part of the task, people, our normal volunteers activate hippocampus. The schizophrenics don't activate anything in hippocampus, and oops, we got a little bit ahead of myself, sorry. Schizophrenics don't activate any part of the medial temporal lobe, and when you look at the normals show much greater activation than the schizophrenics in this area. This is another task, another generalization. This is a generalization task. This is a learning task. This is a simple learning task. And as you can see in simple learning tasks, schizophrenics perform just as well as normals. But in the generalization part of the task, the schizophrenics are, again, deficient. So this performance, these kinds of performance tasks and some of the imaging that I've shown you already have suggested that there is something wrong in the hippocampus. And a number of years ago, I decided to take a careful look at that, since we know really a lot about the prefrontal cortex in schizophrenia and a lot about the deficits in the prefrontal cortex, but the abnormalities in the medial temporal lobe have been more obscure. This is what the medial temporal lobe looks like in vivo. I showed you in a few slides back what the medial temporal lobe looked like concretely as we took it out of the brain, and this is what it looks like. It's about in vivo. The yellow arrows demark the anterior part of the hippocampus, and the black arrows the posterior part. And as you'll hear me say, it seems as though it's the anterior part of the hippocampus that's most abnormal in schizophrenia. Now, this is really just a cartoon of what the glutamate synapse looks like, with a presynaptic synapse releasing glutamate, a postsynaptic synapse, very complex. This is really the least complicated way that anybody could draw this. And the astrocyte. The astrocyte is actually associated with recycling glutamate in this synapse. So for the glutamate system, the astrocyte is really a very vital part of the synapse. One of the additional characteristics of the hippocampus that's very prominent is that the hippocampus is a very excitatory organ. Probably 70% of the transmission, it's estimated, I don't quite know how people make these estimates, but it's estimated that 70% of the transmission within the hippocampus is excitatory. So I want to just tell you about 2 or 3 observations that we were able to make about the hippocampus in schizophrenia, and these observations didn't quite fit together very well. And I suppose it's always when you have to think hard about something and when predictions don't quite turn out like you expect them to, that you might come to the most creative hypotheses. We observed really decreases in NR1 message in hippocampal subfields. So NR1 is the necessary subunit of the NMDA receptor, and the NMDA receptor is one of the excitatory receptors in the brain. And it's particularly, these receptors are particularly dense in the hippocampus. And particularly in the dentate gyrus, we saw decreases in NR1. And that would suggest really, we would interpret that to say that excitatory transmission is decreased if there's a decrease in NR1. Then, on the other hand, we took a look at BDNF message, and paradoxically, we saw an increase in BDNF. BDNF is an activity-dependent protein in the brain. It's a neurotropic factor. And here we see increases in BDNF, both in CA3 and in CA1. Then, in addition to that, we took a look at regional cerebral blood flow in the hippocampus and saw increases, particularly in the anterior hippocampus, in regional cerebral blood flow in schizophrenia. So this, I'll just show you how to read this. The 0 line is the normal volunteer level of regional cerebral blood flow, and this is the percent change in people with schizophrenia in regional cerebral blood flow. And in hippocampus, there was an 8% increase, at least in this particular drug-free sample we were looking at. These people with schizophrenia were all medication-free. And this white line here is represented by this red line. And when we took these very patients and put them back on antipsychotic medicine, we saw a decrease in this abnormality. So what we see repeatedly is that antipsychotic drugs have a big effect in hippocampus. And the effect that they have in hippocampus is an ameliorative or a normalizing effect. So there are two observations we made. One would predict that regional cerebral blood flow, perfusion, neuronal activity would be decreased in hippocampus, and that's because we saw decreases in the NR1 subunit of the NMDA receptor. And then the other observations, this increase in regional cerebral blood flow, and for that matter, the increase in BDNF, would suggest that neuronal activity was increased. So I'm trying to write a grant to get money to fund this, and I wrote in the grant, you know, this is a paradox, and we don't quite know how to explain that. And the reviewers came back again and again and said that they wanted a model for this. And I said, a model? We hardly understand anything about what's happening. How am I going to make a model for this? I remember one time when we had a schizophrenia meeting at Yale, and Pat Goldman's husband, Pascal Rakesh, said to this audience of people, you know, I don't know how you do it in psychiatry. You guys keep talking about hypotheses. And in my field, we just call those ideas. We don't call them hypotheses. But the reviewers, and I understood that in order to get funded for this, I was going to have to come up with a model, so I did. Trying to put together the paradox of the implications that there would be decreased glutamate signaling, particularly in dentate gyrus, and increased hippocampal activity in CA3. So in the process of figuring out what a model that might explain these findings would be, I came to understand more than I ever thought I would about glutamate system homeostatic plasticity mechanisms. So the glutamate system is a system that's really made to affect plasticity. The glutamate system really is in the brain is what we all use for learning and memory tasks. We all use to receive and modify our brain and its activity for environmental probes. So the glutamate system is really designed to be very plastic. So usually, so that there are certain mechanisms that people have described that mediate plasticity. So when signals come into a glutamate synapse, the synapse adjusts in a unique way through plasticity mechanisms to the incoming signals. So there are some times that the whole synapse has to change because of afferent increases or decreases in afferent signals. They have to change without losing the imprinted memory. And there are several kinds of homeostatic plasticity mechanisms that people have written about, synaptic scaling being one, where if you have a code that's already encoded into your synapse, and you have low activity and you want to create some hypersensitivity, or you have high activity and you want to decrease the sensitivity, synaptic scaling is one of the mechanisms that will help preserve the preserve the signal in the synapse, but up or downgrade it. Intrinsic plasticity is another, and metaplasticity is another. And the metaplasticity is the system that I would like to is the method that I really invoked in a model to explain what I saw, in a very testable model, explain what I saw. So in metaplasticity is affected by postsynaptic receptors, both the NMDA receptor and the AMPA receptor. And metaplasticity is the mechanism of shifting the LTP curve to the left. So if you have a synapse and afferent fibers are coming into the synapse, so are decreased, producing some deprivation. What you want to do is you want to increase the sensitivity of your system, and you want to shift the LTP curve to the left so that there's more LTP than there was before. If you have an increase in activity, you want to shift the LTP curve to the right, and there will be less activity than there was before. Now there's both an NMDA mechanism and an AMPA mechanism that mediates this change in glutamate sensitivity, either for the more sensitive or for the less sensitive. If there's more NR2B containing NMDA receptors, the system is more sensitive. If there's more NR2A containing receptors, NMDA receptors, the system is less sensitive. And I'll show you that the way the AMPA receptor sensitizes and desensitizes itself is through phosphorylation of the GLUR1 subunit, its GLUR1 subunit. So there are molecular signals, if you will, there are molecular markers of supersensitivity and subsensitivity within glutamate-rich regions. So the model that I put forward to explain this, and still to this day, I'm not sure I can quite understand why my reviewers liked the model, since it's a very speculative model. I'll show you some of the testing that we've done to test this model, and some of it turns out to be quite interesting. Our idea is that there's a glutamate lesion in the dentate gyrus. No, it's not. It's our idea. It's our speculative model that there's a molecular lesion to reduce glutamate signaling in the dentate gyrus. And what I really think is that there's not just one lesion that reduces glutamate signaling, but there could be a whole family of lesions that reduce glutamate signaling in the dentate gyrus and reduce activity in the mossy fiber pathway, which really reduces afferent stimulation to CA3. And in response to that, a metaplasticity response, one would expect increase in activity in CA3. Because CA3, because there's no because there's no feedback from CA3 to dentate gyrus, there's no way for CA3 to give a message back to the dentate gyrus that it wants it to speed up activity. And in addition to that, the function of CA3 is to be associational, and the recurrent collaterals can become hyperfunctional. So the model really suggests that CA3 has an increase in LTP, and that increase in LTP really accounts for the increase in regional cerebral blood flow. Behaviorally, really, if you were to accept that your CA3 was hyperactive, you might expect an exaggerated pattern completion. You might expect hyperassociational activity within CA3. And one could imagine that if you had occasional lapses or occasional mistakes of association, you could generate the raw material for psychosis or a delusion that if it were formed aberrantly in CA3, that if it were formed in CA3 would get laid down in memory as if it were a normal memory, which would of course account for John Nash's experience that his delusional and his psychotic ideas came into his mind just like all of his other ideas. So I really propose this as a very speculative model, that we would find decreased activity in dentate gyrus because of the decrease in glutamate, that we would find increased neuronal activity in CA3 because of the metaplasticity changes. So now we set out to see if we could really do that. The first thing we did was we did some high-resolution fMRI, and we were using a contrast agent and measuring regional cerebral blood volume. I wanted to show you the image on top so that you can see that with very good high resolution, it won't be it won't be a surprise to many of you here, but if you look at a high resolution image of the hippocampus, you can actually see the area just on the edge, the outer edge of the dentate gyrus. You can see where CA3 is, which is this area right here. You can see where CA1 is, which would be this area right over here, and you can see the subiculum. So you could actually, one can easily by hand draw regions of interest on the hippocampus by subfield. And measure regional cerebral blood volume, which is a perfusion measure. Because we use a contrast agent, the resolution is such that we can actually get real measurements. And what we found, we've completed a whole study now, but not the final analysis yet. This is a preliminary analysis that we did with 4 patients in each of these groups, 4 normal volunteers and 4 schizophrenics off medicine. And what we saw is that in the dentate gyrus, although we don't have a p-value, it's at least a moderate effect size decrease in perfusion in the dentate gyrus, and on the other hand, an increase in perfusion in CA3. So this was some rather concrete support for our hypothesis. Then we turned to, because we wanted to understand what might be the molecular basis of this, we turned to human postmortem tissue, and we decided that we couldn't, I've done many, many measurements of all of the things that I'm going to talk about today in whole hippocampus, and it all of a sudden occurred to me one day that I was just doing it entirely the wrong way, that what we really needed to do was measure all of these molecular markers by subfield. So we now slice 300 micron slices of our hippocampus, and we do dissections like the one that I am showing here. So we looked at the NR1 protein using Western blot analysis, and we again confirmed a decrease in the NR1 protein in the dentate gyrus. In this particular analysis, the dentate gyrus was significant. CA3 and CA1 were not significant. But actually, I must say that in another cohort that I did, CA2 actually also showed a decrease in NR1 protein. So we were able to, this is a full study, so we were able to confirm that, that observation in this study. And in addition to, I told you before that I thought that there was a possibility that there might be multiple lesions, multiple sources of the hypoglutamate function in the hippocampus. And why I said that is that these are the same data that I showed you before on the last slide. But in addition to that, we've done other analyses where we've looked at markers that are on the astrocytes. The astrocytes have all of the they reuptake the glutamate at the astrocyte, take the glutamate, change it to glutamine, funnel that glutamine back into the neuron, and in the neuron the glutamine is changed back to glutamate. So all the glutamate in the synapse is taken up by the astrocyte. It's taken up at the EAT1 reuptake site or the EAT2 reuptake site. So when we looked at both protein and when we looked at fMRI, PCR, we showed increases in the EAT2 reuptake. So if we see increases in the reuptake proteins, it means that the astrocyte is sucking glutamate out of the synapse quite quickly. GlyT1 reuptake site, this is the reuptake site for glycine. And also we found increases in their RNA for the GlyT1 reuptake site. What this would suggest to me is that what we call psychosis and the effect that we see on CA3 is almost a diagnosis like a syndromal diagnosis, like one would make for congestive heart failure, where we can have many different etiologies of a glutamate decrease in the dentate gyrus, and it has the same pathophysiologic effect on CA3, which is to increase region, which is to increase association. Now, let me show you what we find in the in CA3. What evidence there is that would support an increase, a molecular basis for the increase in regional cerebral blood flow. So, the NR2B containing NMDA receptors increase with afferent deprivation, and the NR2A subunit increases with increases in afferent stimulation. So what we find in CA3 is an increase in NR2B in CA3. This is an increase that's at a trend level. This is only 10 postmortem cases, and we've now collected 10 more, so we're in the process of extending these. But what we're predicting that we find is that we'll find an increase in the NR2B, the proportion of NR2B. And when we look, I don't have the data up here, when we look at NR2A in CA3, the normals and the schizophrenics are exactly the same. Now, the other receptor that participates in a metaplasticity increase in sensitivity to glutamate in any particular brain region is the AMPA receptor. And the AMPA receptor is made up of various subunits, the GluR1 subunit being an important subunit. And phosphorylation of the GluR1 unit carries AMPA receptors that are sequestered inside of the cell. It traffics those receptors up to the membrane, so that dephosphorylation of the GluR1 subunit of the AMPA receptor at this particular amino acid, S845, will produce LTP. But phosphorylation of the AMPA receptor at 831 will traffic the AMPA receptor up to the membrane. And what we see in schizophrenia is a significant increase in the phospho-GluR1 in CA3, but not in any other. We've looked at this in all the subunits, and the only subfield in hippocampus where we see these increases in the NR2B and in the phospho 831 GluR1 is in CA3. So our general hypothesis then that we're finding some support for, not of course enough support to call this anything other than a speculative model, is that we have a glutamate lesion in the dentate gyrus in psychosis. My hypothesis would be that what we're looking at is the mechanism for psychosis, not the mechanism for schizophrenia. So that this decrease in glutamate function in the dentate gyrus causing a decrease in activity in the mossy fiber pathway to CA3 really produces an inappropriate sensitivity, supersensitivity in CA3. Now, this inappropriate supersensitivity in CA3 must be contributed to by many different risk genes. In other words, most of the risk genes the strongest risk genes that we know about in schizophrenia, neuregulin, DISC1, dysbindin, are all genes that affect the composition of the synapse, particularly the glutamate synapse. So I'm sure that if this very simplistic hypothesis turns out to be supported, we'll find a lot of different things to look at as risk genes in CA3. So what we're proposing is that we have a lesion in the dentate gyrus that causes a reactive, if you will, supersensitivity in CA3. So now we could really turn towards animal models. We can't make an animal model of schizophrenia because it's just hard to judge whether mice have hallucinations and delusions. But if we can work backwards in a mouse from pathology that we can demonstrate in schizophrenia, if we can work backwards and then look at, then make an animal model that looks like our human model, that would be interesting. So we are actually in the process now of looking at dentate gyrus specific NR1 knockout. There's a particular mouse where you can take NR1 and knock it out specifically in the dentate gyrus. This is another dentate gyrus model, which is an NPAS3 knockout. NPAS3 is a risk gene for schizophrenia, and it was discovered in a Canadian mother-daughter pair that shared the same translocation. And the translocation that they shared, person who was the schizophrenic mother and the person who was the schizophrenic daughter was right at the NPAS3, gene. And then people have really made NPAS3 knockouts in mice and use them as a potential mouse model that would create a susceptibility for schizophrenia. What you can see in this NPAS3 model, it turns out that mice that have this NPAS3 knockout have abnormal neurogenesis in their hippocampus. You can see the difference in the marker. I think this is Ki67, in the marker for neurogenesis in the dentate gyrus in the NPAS3 knockout looks very different than the kind of staining that you get, the kind of Ki67 staining you get in the wild type. And what you can see is that it has implications for the number of cells you can see in the dentate gyrus, in the hippocampal dentate gyrus. Here the wild type and here the NPAS3. And oh, actually, this was BRDU. The number of these BRDU cells is remarkably decreased. So in this particular animal model, the people who were investigating this particular animal model investigated the dentate gyrus very thoroughly, but they totally overlooked looking at CA3. It left the opportunity for us to go in then and look at what happened in CA3. So what we saw in CA3 in these NPAS3 knockout mouse is the same increase in phosphogluR1 that we saw in the schizophrenic brain, although we saw no change in NR2B. Here you see that in the schizophrenic tissue we see an increase in NR2B in CA3 and an increase in phosphogluR3. We're now going on to look at this other mouse model that I had suggested to you before of looking at specifically at NR1 knockouts. So this is our hypothesis that you've seen before, and I guess that I was encouraged by the people who invited me here to speculate on what might be the treatment implications of this kind of a model for psychosis. First of all, one of the treatment implications is that we really switch around our diagnoses a little bit and think of psychosis as a dimension that's important across all of our diagnoses, much like congestive heart failure is a dimension that's important across all different kinds of disease etiologies in medicine. And oftentimes, not always, but oftentimes, treatments for heart disease are directed towards the congestive heart failure element, not necessarily prophylaxis and prevention are directed towards underlying basic etiologies. So one of the things, of course, that if you're thinking that CA3 is overactive, and that's one of the things that may be generating psychosis, because CA3 function is sensitive to the environment. One would think of psychological treatments as being particularly important, cognitive remediation or CBT approaches. And one would have to really demonstrate in animals the extent to which one could modulate, perhaps organize activation within CA3 using CBT or using cognition training. But it would certainly be a reasonable thing to think about. Treatments to decrease excitability in CA3, and those would be things like dopamine antagonists. Dopamine antagonists, although people haven't looked for dopamine antagonist action in hippocampus, our current research would suggest that it's very active, and it's active in decreasing excitability, cholinergic agonists, mGluR agonists. And then there's a group that who's doing inhibitory TMS to the medial temporal lobe. And I just had this is a group, I even forget who the group was, but they reported a striking antipsychotic response when they did inhibitory TMS that was directed to the medial temporal lobe. I just wanted to kind of go through, I think it's getting late, just to show that when we do cognitive remediation in people with schizophrenia, we can change performance, we can increase verbal memory, and we can produce changes in the brain that occur in the hippocampal formation and in the anterior cingulate cortex. So I do think that I don't have any specific, I really have to go about looking carefully at this model to see whether the model is even plausible, and after that would turn towards treatments and would turn towards this kind of a system as perhaps being a marker, CA3 being a marker of antipsychotic activity, which would be interesting. There's many different drugs now. We used to, now that we're really looking at cognitive enhancing drugs, in addition to antipsychotic drugs, I think the idea is that we'll always have antipsychotic drugs, we'll always have psychosis, and therefore we'll need treatments. Now that we're looking at cognitive enhancers, we have a number of new drugs that are more or less coming out of the woodwork that are really designed to enhance cognition and may be very active in the medial temporal lobe system. That being said, I've done this work with many different collaborators, and I just listed out their names. Not that their names may make any difference to you since you don't know them, but they've been my collaborators over time in this project. So with that, I'll end and accept challenges and answer any questions.